أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد المغضوب عليهم ولا الضالين
Good morning everyone and Assalamu alaikum. How are you today? I am teacher Aynah and welcome to this new day of exploring ideas and acquiring knowledge. I believe that you are now in your comfortable place of learning, so stay at your seat and keep watching. I am grateful that all of you are present today class and I can feel that you are all excited. But before we formally start our lesson, let's have a review first of what is genetics. Very good. That is the definitions of genetics. And today, we will explore our first lesson in General Biology 2, the Mendel's Law of Inheritance. This topic is interrelated to Practical Research 1, Pre-Calculus, General Math, Reading and Writing, Statistics, and Probability. Are you ready to learn? Come and join me as we explore another learning experience today. We will start our lesson for today by doing this activity. I would like you to watch attentively, listen carefully, for you will be asked some specific questions. Have you ever been dreaming on superpowers, like superheroes? It was the fantastic dream during teenager's life. Would you like to be a scientist or maybe study about inheritance of trait? What will be your first experiment in genetics? Do you know who the father of genetics is?
Monk, teacher, and biologist, Gregor Mendel is known today as the father of modern genetics.
Gregor Mendel's experiments on pea plants were fundamental in establishing the field of modern genetics.
Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic, Mendel excelled at school and became an Augustinian monk to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the university in Vienna, and that gave him the education in science, math, chemistry, and botany that he then brought back to use in his teaching.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments.
Before Mendel, people people thought you would equally inherit the traits of parent A and parent B in equal proportions.
Mendel experimented with pea plants and found that instead there were dominant and recessive traits in all organisms that were passed on statistically based upon the mixing of genes between the two parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly and are independent of the other traits and of the traits of the two different parents.
The ability to explain hereditary diseases and to engineer plants with desired properties are all consequences of the fundamental theory founded by Mendel.
During his life, Gregor Mendel's research was disregarded by almost everyone. It wasn't until the early 1900s, after his death, that Mendel's work was appreciated and now serves as the basis for genetics and heredity.
In his later years, Mendel became the abbot of his monastery and spent less time with his scientific endeavors. Gregor Mendel died in 1884 at the age of 62.
Gregor Mendel's work laid the foundation for modern genetics, for modern gene therapy, and is the basis for some of the most exciting discoveries in medicine currently and in the near future.
Can you list down what are Mendel's laws of inheritance? What subject major that Mendel studied? Get your paper and pen and write your answer.
Very good. You know already the story behind on Mendel's experiment. Before we further discuss about the Mendel's experiment, we first define the following important terms.
Gene, a unit of heredity, is a section of DNA sequencing encoding a single protein. Alleles, two genes that occupy the same position on homologous chromosomes that cover the same trait. Dominant is the allele of a gene that masks or suppresses the expression of an alternate allele. The traits appear in the heterozygous conditions. And recessive, an allele that is masked by a dominant allele does not appear in the heterozygous condition, and it only appears in homozygous conditions. When we say homozygous, is a having identical genes for a particular characteristics, while heterozygous is having two different genes for a particular characteristics. Genotype, the genetic makeup of an organism's. Phenotype is the physical appearance of an organism.
In Mendel's laws of inheritance, he used Punnett square to identify genotype and phenotype of a trait in monohybrid and dihybrid cross. For us to know the Punnett square, let's watch this video.
Thanks for stopping by. Today we're talking about the basics of Punnett squares. Punnett squares measure the probability or likelihood of a genetic outcome based on a specific genetic cross. It doesn't tell you what will be, only what could be. For the simplest of Punnett squares, you'll cross two genotypes. These parent genotypes will be for the same gene but may or may not contain different alleles. Let's look at an example of a genetic cross for eye color. Green eyes being dominant and blue eyes being recessive. And we'll use big G, little g for the maternal genotype and little g for the paternal. So this is our cross. The mother has green eyes and the father has blue eyes. And we want to see the probability that their offspring will have either green or blue eyes. For this cross, we'll draw a square split into 4 quarters, like a simple 4-panel window. This is the Punnett square. One of the parental genotypes will go on top and the other will go on the side. It doesn't matter which genotype goes where, and the order of the letters really doesn't matter, but we conventionally write the dominant allele first, if there is a dominant allele. So your setup should look like this. Then you simply carry the letters through to their respective quadrants and match them up, like so. Right, each quadrant will represent a 25% chance that that genotype will result in the offspring. So in this case, we have only 2 possible genotypes, big G, and little g, and each genotype has a 50% chance of showing up in the offspring. Similarly, you have two possible phenotypes, green eyes and blue eyes, with a 50% chance of each. Let's look at another example. We'll cross two genes that are heterozygous for eye color in this example. So big G little g crossed with big G little g. These colors are just for illustration purposes. So we again will draw our Punnett square and put one gene on top and the other down the side, and then carry the letters through to their respective quadrants and analyze the results. So this cross results in a 1:2:1 genotype ratio, with 25% homozygous dominant, or big G, 50% heterozygous, or big G, little g, and 25% homozygous recessive, or little g. The phenotypic ratio would be a 3:1 ratio, with 75% green eyes and 25% blue eyes.
And now, you already understand how to use Punnett square. Again, these are the steps in using Punnett square. First, draw Punnett square. Next, write genotypes of the parents. And step 3, fill in the boxes. And step 4, determine the percentage of offspring. Now, get your paper and pen and let's try to solve this one.
Activity number 1. A blue heterozygous flower is crossed with a blue heterozygous flower. White is the recessive trait. You can use a Punnett square to determine the probability of one of their offspring having a white color. Identify the genotypes as well as identify the phenotypes.
For your group performance composed of 3 members, interview a family from your barangay that has an observable trait and try to use a Punnett square to predict the inheritance of their children. Try finding out the probability of each child to get the observable trait from their parents. Create your own allele to identify the trait. Good luck! Now, let's self-identify Mendel's conclusions. Number 1, inheritance is determined by chemical factors that determine traits are passed from one generation to another generation. So, these chemical factors are called genes. Number 2, principles of dominance. Some alleles are dominant while others are recessive. And dominant allele always expressed unless there are 2 recessive alleles. And now, to be sure how far you have learned, I would like you to get your pen and paper and let's do this. We will answer a 10-item quiz. I will be posting the questions on the screen and you will be given 10 seconds to think of your answer, and I will reveal the correct answer before we will proceed to another.
Very good! I can see that you really understand our lessons for today. So for your assignment, I would like you to research about the genetic engineering. What are the importance of genetic engineering in our country, and what are the latest trends of genetic engineering?
and I hope you get all of my instructions. So it has been a great day to all of us, for we have learned something new. And it also proves that even COVID-19 can stop us from learning. So once again, I am Teacher Aynah, and see you again for another episodes of learning. Learning in science is always fun.